Hello, I'm Matthew Malcolm with American Vineyard Magazine, and today at the Central Coast Grape Expo, hundreds of growers gathered here at the Mid-State Fairgrounds in Paso Robles to learn the latest in pest and disease management, among other seminars that took place, and a trade show. One of the featured speakers today included Houston Wilson, PhD at UC Berkeley, who discussed strategies in improving biological control of leafhoppers in the vineyard. Here's Houston.
Biological control with generalist predators is certainly possible, and in many cases has been demonstrated to be very effective. But in other cases, it's a lot more, it can be very problematic.
What we've did over a 5-year period was a project that was in collaboration with numerous North Coast wine grape growers. And these growers approached us initially to evaluate the use of flowering summer cover crops as a means to attract more beneficial insects, which could then increase biocontrol of grape leafhopper in particular. The first stages of this study was to actually identify the flowering cover crops that would work in commercial vineyards. So we tested a number of different flowering cover crop species, sweet alyssum, buckwheat, California bluebells, wild carrot, bishop's weed, various clovers, you name it, anything that folks were interested in that they thought might work in a vineyard, things that we saw from the scientific literature and other studies that had been used to enhance biocontrol in vineyards in particular. And so for 3 years we had a number of split plot trials looking at the insects that were coming on to these flowering cover crops. We were doing a lot of grower exchanges to talk about equipment, seeding rates, how to mow them, etc. Just how to manage these things in a vineyard. And at the end of that 3-year period, we arrived at the use of 3 species of summer flowering cover crops. Phacelia tanicetifolia, I think it's called purple tansy is the common name, Amymajus, bishop's weed or bishop's flower, we call it, and then Daucus carota, which is wild carrot. So these 3 species of cover crop were selected because they had an overlapping bloom sequence, the Phacelia blooms, then the Amy blooms, then the wild carrot blooms, all in a row over more or less the entirety of the grape growing season. Second, the flowering cover crops were easily integrated with vineyards. They were sown in the fall. A lot of folks said that was very important because they already had their seeding equipment out. They were putting in overwintering cover crops. It was easy enough to throw those flowering cover crops into the hopper and sow them in the vineyard as well. All the species of flower were rain-fed. They don't require any supplemental irrigation, which is of course very important in California. And then of course they attracted beneficial insects to the flowers themselves. So what we did after identifying this cover cropping program, or at least working it out with growers, we went ahead and did a 2-year study where we had plots with and without the flowering cover crops at 10 different vineyards throughout Napa and Sonoma County. The flowers were sown all together in alternate rows over a 1 or 2 acre block, depending on the size of the experimental vineyard. And these flowers, they come up sequentially. So the Phacelia comes up, it blooms, and then the grower would go in and mow it, and that would keep it from going to seed and spreading underneath the vine row. The next species of flower, the Amy, would come up after that, and similarly you would mow it after it blooms, and then the wild carrot comes up, mow it after it blooms, and then till the whole thing under and go in to pick the grapes. So the results of this 2-year study, what we found was that the flowering cover crops at peak bloom, they had a much higher abundance of beneficial insects relative to the control plots, which just had, weedy vegetation or a low mown, permanent grass cover, something like that. So the flowers definitely attracted more beneficial insects, but what we subsequently saw was that never translated into increased beneficial insect populations in the canopy of the vine itself. And subsequently we didn't see any increase in parasitism rate. We never saw any decrease in leafhopper populations in the presence of the cover crops. So they really didn't seem to enhance biocontrol. They brought in a lot of good bugs. That didn't have a knock-on effect in terms of actually augmenting biocontrol, reducing leafhopper populations. But during the analysis of this this data, we did see that there was a lot of variation in leafhopper populations and in parasitoid populations across the different research sites. And that made us think that there maybe was something specific about the landscape that these experimental vineyards were located in that may be driving pest and parasitoid populations. And we thought that this was particularly likely with grape leafhoppers and their anagris parasitoids, because the anagris parasitoids have to leave the vineyard during the winter to find an alternate host in order to successfully overwinter. We typically find these hosts in natural habitats outside of the vineyard. And then in the spring, the anagris migrate back into the vineyard to continue reproducing and attacking the grape leafhopper. So these anagris overwintering sites, if they're far away from the vineyard, it's really going to affect the timing and the abundance of the parasitoids moving into the farm. So we actually kind of took a snapshot around each vineyard and quantified how much natural habitat was around the farm, and then used that as a variable to analyze parasitoid populations, leafhopper populations, etc. And we did see that vineyards that had higher diversity landscapes, more natural habitat around them, had more anagris earlier in the season. This led to increased parasitism of leafhoppers and ultimately lower populations of leafhoppers at the end of the season, which in the North Coast, this is really when a lot of growers make their decision about whether or not to spray for grape leafhopper. So we then went on to identify the plants in the natural habitat that the anagris were actually overwintering on. It's not just any natural habitat is equally beneficial, but rather what plants specifically are acting as host for the parasitoids during the winter. And we found coyote brush, blackberry, in particular, to be really the primary host. There's a number of other plants that the parasitoids utilize, but they're things that are found in gardens or hedgerows, like roses or sage or mint plants, and you could potentially plant those around your farm as well, but it's something that doesn't naturally occur in the landscape of Napa and Sonoma County. And then finally, we did another study where we looked at vineyard proximity to riparian habitats to see if there was any effect on biocontrol, given that there's a lot of blackberry typically in these riparian habitats. What we found was no trend in terms of beneficial insect populations or their impact in terms of parasitism. There's really no difference between the vineyard edge close to the riparian habitat relative to the vineyard interior. But we did still see a difference in leafhopper populations between the edge and interior. They seem to be laying more eggs at the interior rather than at the edge. And when we lined this up with grapevine vigor, it was measured by total petiole nitrogen content, we did see that vines that had higher vigor had increased leafhopper deposition. And the vines with higher vigor were typically at the interior of the vineyard. And so what we were seeing is that the riparian habitat did seem to have an effect on the pest populations, but it seemed to be due to the fact that the riparian habitat, when it's adjacent there to your vineyard, there's changes in microclimate, increased competition by having vines next to these big perennial trees and things that are in riparian habitats, that was leading to lower vigor and thus lower leafhopper densities on the vines themselves. So kind of the big picture out of all this, and some of these findings, like the relationship between leafhoppers and crop vigor, is something that's been shown in other studies before. That's not necessarily new, but I think what's really unique from our work is the identification of the overwintering habitat that the anagris utilize. There's some plants like coyote brush that we previously didn't know was an overwintering host, and it's all throughout the North Coast. So if it's a grower who's looking to manage leafhoppers by using habitat manipulation, really it seems like the best approach would be to increase coyote brush and blackberry and these other overwintering host plants around the farm. The area needed relative to the area of overwintering habitat relative to the area of vineyard is still a big unknown, but we've at least gotten the first step in terms of identifying what plants these parasitoids are overwintering on. The flowering cover crops really didn't seem to do anything in terms of biocontrol, so that's something that you may not want to spend time and resources on putting into a vineyard. The relationship between crop vigor and leafhopper densities, again, not unique to our study, seen in a number of other vineyard studies, so that's something you want to keep an eye on, maintaining, moderating vigor, so overly vigorous vines are going to attract a lot more leafhoppers. And then some of the stuff that we weren't specifically doing research on, but other cultural practices that can reduce leafhopper populations would be to really pay attention to the timing of canopy thinning, suckering early in the season, because the leafhoppers, as they emerge in the spring and start to oviposit and feed on the grapevines, they will oviposit and feed on a lot of those suckers and a lot of that basal foliage that may be removed later in the season. So if you can time that removal of that vegetation to take place after there's been some oviposition from the leafhoppers, after we've seen them active on the vines for some period, that could be a way that you can reduce the overall density of eggs on your vine. You're pulling off these suckers that have leafhopper eggs on them, and ostensibly that's going to kill off the leafhoppers there.
Thank you, Houston. Gain more of the proceedings from today's event by reading American Vineyard Magazine, along with watching the other video reports related to this year's expo. I'm Matthew Malcolm, CaliforniaAgNet.com.